So let's get started. I will give you an overview of the past, the situation today, and a little bit looking into the future about cytotoxic drug exposure. But I have also decided to add another group of drugs, antibiotics, to my presentation. Because antibiotics is rather new when it comes to dangerous activities, exposure, and things like that in healthcare. And it's popping up the last 3 years, roughly, that antibiotics are maybe also, giving at first health effect when it comes to exposure. So I have put that into my presentation too. This is my background. I am not a nurse. I'm not a technician, pharmacy technician. I'm not a pharmacist. I'm a chemist. So I'm very interested in molecules and what they do and how you can produce them and how you can manage them. But I started in 1980 with my study chemistry, and at that time it was very usual that students were very laying back, so it took me 8 years to finish that. Nowadays it's not done, but that time it was a good time. Today it's maybe 4 or 5 years, but that time I was fine with that. So then I started my PhD. It was sponsored by the Dutch Inspectorate of Labor of the Dutch government, and it was the aim to develop test methods for validation of exposure to cytotoxic drugs. I finished that in 1993, and then later on, the university gave me the opportunity to make a business out of my PhD study, which I did, and I founded in 1995 Exposure Control. It's a company, a consultancy who was involved in monitoring exposure to cytotoxic drugs, and we do that nowadays around the globe. In 1996, I finally got my PhD. So this is a little bit about my background. The agenda for the cytodrugs today, there are many issues to address. Some will be a little bit more in detail, others will be more in an overview. Talk about toxicity guidelines, European directive. I know European is a little bit controversial nowadays here in the UK, but anyhow, you're still in the European Union. So occupational exposure, sources of contamination. When you work with cytodrugs, where the contamination comes from, what is happening, where are the leakages, where are the problems, things like that. How do you protect yourself? Monitoring. Monitoring is actually, when you work with the drugs, you can monitor if you are exposed, you can monitor if the drugs are in the environment, so are there many options to do. I will focus much on surface contamination, a little bit about the references, what does it mean when you have these data. Also talk about exposure, real exposure of workers by urine testing. So if you do a urine test, you can see if people are really exposed. That's available nowadays. Closed system drug transfer devices, very hot issue nowadays. Cleaning, and I will end it with some conclusions, recommendations. That's the first part, and then the antibiotics will come. I have a question for you. This is the first one. So let's try if it's working. Which of the following statements are correct? Most chemotherapy drugs are carcinogenic. Most of the chemotherapy drugs are reproductive toxicity. Most of the chemotherapy have both, actually, there's 1 and 2. Most chemotherapy drugs are hazardous drugs. Or 5, all hazardous drugs are chemotherapy drugs. So it's up to you. Please press 1 till 5 what you think is the right answer. Some to go. I have only 50 votes now. So these are the results. So you have voted 44%. Most of the chemotherapy drugs are carcinogenic and reproductive toxicity, and 4 is the 53% said most of the chemotherapy drugs are hazardous drugs. The correct answer is 4. Most chemotherapy drugs are hazardous drugs. That's the right answer. Actually, 3 is quite okay too, but it's a little bit more than that. Here is the toxicity of the drugs as effectively shown in a previous slide where you answered on. We have to deal with 2 effects when it comes to cytotoxic drugs. We have acute effects. Acute effects are effects that actually are found in patients. When patients are treated with cytodrugs, you get all these kind of effects, and sometimes it can be very severe, sometimes it can be quite okay. That's on the left column, acute effects. It's high dose, caused by high doses given to patients. The right column, the delayed effects, that are effects that happen after probably 5 years or 10 years or 20 years. That are effects you don't really see that much in patients. It happens there, but not that much. But it's more effects that are related to occupational exposure. So for pharmacy technicians, nurses who are working with cytodrugs day by day, by day, by day, after a certain period, there is an option that one of these effects happen. Reproductive effects, that is seen. There are studies published about it. Spontaneous abortions happen. Low birth rate is often seen. Prolonged time to pregnancy is often found. So it's really related to exposure for nurses, technicians who work with these drugs. And the other thing is carcinogenicity. It's very strange, but when patients are treated for cancer, sometimes secondary cancer appear after 5 or 10 years. And therefore, the idea is behind this that carcinogenicity is not really linked to only the exposure, high exposure levels in patients. But if you are exposed day by day by very small amounts, then there can be a risk for workers too. And actually, when you go to the classification, there is a classification on carcinogenicity, then you will find this classification by the International Agency of Research on Cancer in France. It's a part of the United Nations Association. And there it's indicated that all type of chemicals, but also cytotoxic drugs, can be divided in several classes. And class 1 is actually the class with the most dangerous drugs, because from these drugs or these compounds, it's known that they can really cause cancer. Then we have class 2, A and B, and these are drugs and chemicals that have been shown in animal studies that they can cause cancer in animals, and they cause cancer in animals, and therefore are probably also causing cancer in human beings. That's 2A, 2B. Then you have class 3 and class 4. So if we go, for instance, to class 1, we know this is just a few examples that these, let's see, 7 drugs are known carcinogens. So they're really the class 1, the most dangerous one. And I think when you take cyclophosphamide, etoposide, everybody knows that they are used frequently nowadays in hospitals. The others probably not that much, but cyclophosphamide, etoposide are really frequently used. When we go to class 2A, here are 4 types. I think cisplatin, doxorubicin are frequently used today now in hospitals too. So these are also carcinogens. Then we have class 2B. This is another example. Mitomycin, well known for bladder installation, is class 2B, carcinogenic to humans. Daunomycin, dacarbazine are not that frequently used. But anyhow, showing that it's a large group of drugs who are carcinogenic to humans and therefore also can cause cancer in people who are exposed to very low amounts day by day by day after 10, 5, 20 years. This is a very relevant sheet. Actually, it's talking about a specific group of carcinogens, so chemicals and drugs that can cause cancer. And it's especially the group class 1. The class 1 group, from these chemicals and drugs, it is known that when you get these in your body, that a very, very small amount can have an induction on your body to cause cancer. And for these drugs, it's very relevant to protect yourself in a very good way. And actually, from this information, it is decided that it's not only for these drugs, but it's for the total group of cytodrugs that you have to have that protection. which means that you have always to avoid exposure. So never get the drugs in your body or on your skin or whatever. You have to be well protected, therefore you take all these precautions nowadays. We have the guidelines, we have protective measures, we are working in isolators, we have cabinets and all these things. And in the end, there is also an obligation that if you work with these drugs and there are methods available, then you have to really check the workers if they are exposed or not. So, in fact, if you work with these drugs, like cyclophosphamide, which is one of the most, I would say, toxic one, but also cisplatin is also included in this group. If you work with these, then you have to be very, very much on the safe side. So that's the situation when it talks about a very specific group of carcinogens. Safety levels are described in the European Union Directive on Carcinogenic Compounds as 1. Obliged in a random order. 2. Obliged in a hierarchic order. 3. Advised in a random order. 4. Advised in a hierarchic order. Or 5. They are not present. Please give your answers. This is a tricky question. I have 50 votes now, I'm missing some. Well, 54, 55, it's coming. Okay. Good, okay, we will see. Oh, this is interesting. Quite a lot of opinions. Let's start with the majority. The majority said 42% advice in a hierarchic order. And the second group said obliged in a hierarchic order. So actually, the total group is saying there is some hierarchy in how to handle this, which is in fact true. The only thing is that every directive coming from Europe is not seen as a law. So that means that every member state within Europe has to make its own law and rules on a directive. And that means actually that the directive is not obliged, but it's advised in this case. So actually answer 4 is the right one. But it would be good that answer 2 would be the future, because that will improve your safety. And now I'm fine with these questions. So quite good. So when we go to that directive, which is already from 1990, so it's long ago, and there has been some updates nowadays. But anyhow, we go back to the past 1990. There you see what the strategy is within that directive. And it's about carcinogenic compounds, but as you know, most of the cytodrugs you work with today are also carcinogenic, so it applies for these drugs too. So the strategy is very simple. First, if you work with carcinogenic compounds, then you have to replace it by another one if it's possible. If it's not possible, then you go to the next step, and you have to reduce the exposure. So sources of exposure. So you make barriers between the drugs or the compounds and yourself, so you're not getting in contact with it. If you cannot do that, then you have to work on ventilation. So if drugs are in a room or whatever and you create enough ventilation, the drugs or compounds will appear. So they will disappear, sorry. They will disappear and get away from you. And finally, if all these things are not possible, you put on your personal protective equipment, your gloves, your gowning, all the things together. But that's actually the last step in the total strategy. If we apply this on carcinogenic compound, but then for the cytodrugs, so in practice for you who work with the drugs, then the replacement is not possible, because when a patient needs cyclophosphamide, you cannot say, well, this is not good for my safety. You get 5-FU because it's better for me. But that's not done, of course. So it's not really an option to change the drugs. So step 1 is impossible in oncology. So then you have to automatically go to step 2. And that means that you have to make the barriers between you and the drug. And nowadays there is only one option to do that, and that's working with closed systems. Closed systems is the only option that you make a barrier between you as a worker and the drug to get in contact with the drug. So it's a real barrier. It's closed behind you and the drug. Ventilation, that's what we've seen in many hospitals nowadays, is that a lot of hospitals work in room. You have clean room facilities, you work in isolators, you have cabinets, you have in the nursing areas ventilating the rooms and things like that. So ventilation is another option to get away of the drugs. But of course, there are many settings where there is no ventilation. And I will come later on that when it's about antibiotics. If you have no ventilation, but you have spill and leakage in your room, and you don't clean it, you don't remove it, then it starts to evaporate. So particles are getting around, you inhale it, or you get in contact with it. So if you have a bad ventilating room, for instance in nursing departments, which often is in very old buildings where you have no air conditioning or whatever, then it can be that contamination of drugs is increasing if you don't take it away. And in the end, what is done very often nowadays, people have a lot of things on their body, like the gloves, the gowns, mask, clothing, shoes, and whatever you can think about. But actually, that is the lowest way of protecting yourself. You have to do it, but you have to go higher up in the hierarchy. Actually, the first step now is closed systems, then ventilation, and finally, you get the personal protection. So when we look in Europe, and I think it's probably the same here in the UK, level 3 are mostly applied in Europe, and some hospitals apply level 2. So it's a really step to go forward to have implementation of closed handling. Okay, that was the directive, 1990, long time ago. It was the time that I started with my first study, so it's already a long time ago. But the directive will be upgraded and updated over the years, and they are working on it now to have also the mutagenic and reproductive drugs in that directive. So it will be a very comprehensive directive in the future. Transfer of contamination with cytotoxic drugs is most likely by 1, skin contact, 2, ingestion, so you eat it more or less, inhalation of aerosol and particles, 4, all of them in unequal share, 5, all of them in equal share. Go ahead. It seems an easy one because everybody is responding so quickly. So. Okay, let's see. 81% said all of them in unequal share. And actually that's the right answer. Because the exposure is on many ways in potential, and it all depends on how the way you are dealing with the drugs and protect yourself. But really relevant is, I will come later on it, that skin contact is very important. But it's, Good. You are well, well trained, well informed. Great, great. Okay. Occupational exposure to the cytotoxic drugs. It's linked to the question before. There are, in fact, in general, 3 options to be exposed to whatever drug, whatever compound. But for the cytodrugs, I will specify a little bit more. Inhalation is an option for particles and vapors. We know that some of the cytodrugs can evaporize. So, for instance, if you have cyclophosphamide, I take it as an example, if you have cyclophosphamide leakage on a table or surface or whatever, we know that it starts to evaporate. So probably when it's on the surface and you come back a week later, then it's not there anymore. It just evaporates. So if you are working in such an area, then you of course can inhale the drugs if you don't protect yourself. That's an issue. The second is ingestion, and actually ingestion is that you eat the drugs. Well, how can you eat the drugs? There are a few options actually. I think there are mainly 2. If you eat and drink in an area where these drugs are, which is actually forbidden, you don't do it. That's an option. But sometimes what happens, and people will recognize, is that you can touch things and then put sometimes your finger on your lips or whatever. And then we call it finger shunt effect. Then you can, of course, ingest it. But that's not so very common. Most relevant is the last one, absorption through the skin. And it has been already known for, I think now 15 years, that this is the major exposure route. And it's quite very simple. When you get in contact with the drugs, and especially when you take drugs or surfaces or touch surfaces, or you take vials or whatever in your hands, and you don't protect yourself, it's very easy that the drug is on your skin. You don't see it, but it can pass your skin very quickly. Good example of it is cyclophosphamide. I call it again. I will come later on every time on cyclophosphamide as the worst case when you talk about cytodrugs. When cyclophosphamide is on your skin, in about 15-20 minutes, 80% of the drug is already in your body, and you cannot take it away. You cannot wipe it away by washing your hands or whatever. So when you touch this drug or vials or whatever, it's very dangerous if you don't wear gloves. So it's really important. It's a very strong recommendation. Whenever you get in touch with drugs, or think you get in touch with drugs, even when you open packagings or whatever, where drugs and vials are in, always use gloves. You don't need the thicker ones, the very expensive one, but just a thin one can be okay too, just to protect your hands. So whatever you touch and get in your hands, protect yourself. So be aware of that, because the drugs can be very quickly in your body. So the final uptake of this whole process depends actually on a combination of these routes. Do you have a good ventilating area? Then probably skin contact is the most relevant. Have you a bad ventilating area? Then inhalation can be an issue too. But mostly it's a combination of these 3 routes that gives the exposure if you are exposed. So, and then of course you have your personal protection. So if you use gloves, then you make actually a barrier between you and the drug. So if you get in contact with it and you have it on the gloves, you can throw it away and you are safe with your hands. Okay, so that's the main occupational exposure sheet, how people get exposed to the cytodrugs, but skin contact is really, really the most relevant one. Well, now we look in detail where is the contamination coming from. We know when we do testings everywhere around the globe that contamination is in pharmacy, contamination is in and around cabinets and isolators, and contamination is on the wards, outpatient and outpatient situations. So drugs are there, but where is it actually the most predominant? Where do you find it mostly? I put I focus on 2 situations. The pharmacy. Well, the pharmacy, there are 4 main issues which are relevant. It's the vials. It's the spillage during the preparation technique. So if you prepare, there can be spillage or leakage. So it depends how you handle. And also it depends who is handling, because there are big differences between persons who handle the preparation. The biological safety cabinet, or the isolator, and waste. That's the other point. If we go more in detail, for instance, the vial contamination, it's known by many tests done over the years that vials with cytodrugs on the outside can be contaminated. The very simple reason is when the industry is producing the vials and filling these vials, at the end of the process, when the vial is ready, they wash it. But they are not allowed to wash on the top of a vial where you have to flip off. There you cannot wash the vial because there is a potential risk that drugs or water by cleaning is coming into the vial. So it's not allowed. So they wash only the glass part and the bottom part of the vial. So be aware when you have these vials and you touch it and you take it in your hands that they can be contaminated. So when you touch vials, use gloves. It's known that many of these vials are contaminated. And also the packaging, the carton packaging. Inside, when it's on the vial, it will also be on the packaging. So when you open packaging, always have gloves on. The handling technique is also very relevant because you can realize when you have a vial and a needle syringe and you put a needle in the vial, you take it out, you have the over-under pressure problems there. So you have leakage there. So if you do it in that way, there's always the potential risk of being exposed and get contamination. The cabinet, the biological safety cabinet, and the isolator are in fact designed for protection of the drug and the preparation. They are not designed for your safety. A lot of people think when I work in a cabinet or in an isolator that I'm working in a safe environment. If you take a cabinet with an airflow and you put your hands in, you always disturb the airflow, so it's always airflow going out. So you can be exposed to that. It's the same with an isolator. If you open the isolator at the end to take products out, you can be exposed to there. So isolator and cabinets are not designed to protect you. They do a little, but they are not designed for it. And then we have the waste. In the end, we have a lot of cyto waste. We have the vials, we have the syringes and all these things. And what's seen many days now, still seen nowadays, is that when it's thrown in a waste bin, where you have these, how do you call it, you put it with a pedal and then you get this effect. So if you open it that way, then you get a turbulence therein and all these drugs are coming out. So if you use this kind of systems, which are in fact totally open systems, then you get a lot of drugs getting out and you get, of course, exposed. So you get it on your skin, you get it on your face. So it's really not done to use these things anymore. And there are devices who can save it and seal it actually, so you need not to be exposed. So very simple solutions for this. That's the pharmacy. Now we go to the ward in an outpatient nursing. And well, there it's a little bit more complicated. And the reason is very simple, because in the pharmacy we work with bottles. And in the nursing, we work with patients. So that's totally different. And a vial you can handle and you can manipulate in the way you want. But a patient is more problematic. A patient is, it wants to move and has a lot of drugs in his body, her body. And so it's not so easy. And I've seen in many hospitals that patients' departments are spread, where oncology drugs are used, are spread around in a hospital. So you have many departments where these drugs are used. And it's more policy nowadays to concentrate this all together on one department or probably two to have control over potential contamination and exposure. So it starts in the ward, inpatient, outpatient nursing, where all these prepared drugs are coming from the pharmacy. And I think here in the UK, too, then in general, pharmacy is preparing all the cyto drugs. And then it depends, of course, how they do it. And of course, there will be a lot of pharmacy departments who do it perfectly and whatever. But in general, we know that a lot of bags with drugs coming to the wards are contaminated on the outside. Very simple. In an isolator where you do a preparation, you will always get drugs there. It's getting on the bag. Of course, they try to clean, send it to the ward, but it's always the case, or mostly always the case, that these bags get contaminated. So if you touch them, have gloves on. Then you have, of course, the patient. The patient is a very, you work for the patients, of course, but the patients, I call it always a bomb. The patient is a bomb with drugs. And wherever the patient is going, there will be drugs. So if the patient takes a shower, there will be drugs in that room. If a patient is going to the toilet, there will be a lot of drugs there. If a patient is moving to a relaxed area or whatever to meet family members, there will be potential contamination. So wherever the patient is moving. And then you have, of course, the linen, the clothing of the patient with a lot of sweat inside, probably urine. It's all be there. So if you take the linen from bed and you put it this way, then you get all the aerosols with the drugs going around. So you have to be really careful when you do it to avoid a lot of circulation of these potential drugs in this bed linen. Then we have to deal with the waste. Actually, there's the same problem as in the pharmacy. You have a lot of these waste bins like this, and you see all these drugs coming around. You don't see it, but it's there, I can assure you. So take care of that too. And of course, as I said already, the patient's clothing and laundry. That's another issue to think about. So there are a lot of things to manage. And of course, you cannot do it all perfect, I know, but it's good to know where the pitfalls are, where the problems can be. Which of the following engineering control systems can be labeled as closed systems? And I know you know, as I said already, what closed systems are, so they protect yourself. It's a barrier product. Well, 5 answers again. Biological safety cabinets 2, type 2 and 3. I'm not sure if you use a lot of cabinets here in the UK, but probably. Negative pressure isolators. Positive pressure isolators. All of them or none of them. Okay. 68% voted for none of them. And 70 said all of them. Oh, that's a little bit different. Okay. Well, actually the answer on the question is 5. None of them are closed. So I hope that I can convince the 17% who said all of them, that it's none of them. Actually, cabinets, as I said before, is an open system. An isolator is also an open system. So at the end, you have to open to get the drugs out, and it's the same with a cabinet. So they're not closed. Protection against exposure to cytotoxic drugs. Well, there are, in fact, I would say 4 main things you can do to protect yourself. And it's not only what you do, but it's also what the government, what state, what Europe is doing. And the first step is, I would say, a lot of paper things. Directive guidelines and things like that. So a lot of paper things you can follow to protect yourself against cytotoxic drugs. And we have, of course, legislation here in the UK. You have international guidelines. So there are much information you can gather about how to handle in a safe way cytotoxic drugs. The second is administrative controls. And that's, of course, also a little bit of a more or less paperwork. And that's following protocols. You have protocols for preparation, you have protocols for administration, probably protocols for cleaning and all these things. The third is, then it comes really to practice, is engineering controls. And actually engineering controls makes a barrier between you and the drug. And there we have three general options, I would say. That's clean rooms, working in clean rooms, using ventilation, as I said already before, and you have the cabinets and the isolators. That is more or less the engineering controls you have. And then in the end, you have your personal protective equipment. So how you put things on your body to protect you to be exposed. Well, the actual situation in the US is a lot of variety among these ways of protection. And there are countries where you have probably a cabinet or an isolator, but where the protection of the workers is only gloves or sometimes even no gloves. And I think here in the UK and I think northern, middle and south of Europe, then it looks quite all okay when it comes to personal protective equipment. But when you go to the east of Europe, then you see sometimes situations which would be the 70s and the 80s here. So there is a lot to do there. But so it depends a little bit on the country, the hospital, and how people protect themselves to cytotoxic drugs. Personal protective equipment, PPE. Well, there are many things how you can protect yourself. I think most common nowadays are special clothing, gloves, masks, and goggles. I think that's the main thing you see nowadays. Hair protection, special shoes. Well, you can do a lot. There are many things you can do. But what is relevant actually that the PPE is always additional to the previous things like engineering controls, administrative controls. So you have always to do other things first, and in the end is the final step that you take PPE. And that's a very standard procedure in occupational hygiene. So when you're dealing with chemicals or with dust or whatever, the standard occupational hygiene strategy is that you take always PPE in the end. You have to do a lot before to prevent exposure, and if you expect it or you think there may be an accident or whatever that can happen, there you have your PPE for. That's the standard procedure actually. And nowadays a lot of people focus very much on PPE. The more PPE you take, they think the more safe you are, but that's probably it is, but it's better to prevent that you get exposed. Okay, in the end, when we know that people work with these drugs, and we know that sometimes people smell the drugs. In the end, you have the opportunity to do monitoring. And monitoring is actually what you can do in two ways. One way is that you check in the environment. So the working environment can be the wards, that can be in the pharmacy department, can be in clean rooms, can be everywhere actually. And the other thing is that you look into the worker itself. So you go into the body of the worker and check if the worker is exposed or not. When we go to environmental monitoring, which is indicated in the left, then we check the environment, but we get no information if people are exposed or not. So for instance, if we check contamination on surfaces or wherever and we find contamination there is no indication that that contamination results in exposure because people can be protected well and so on. So it doesn't give that information. And we cannot make a risk assessment. So that's the point, good to know when you go to environmental testing. But a lot of people say, well, I'm fine with all this environmental testing. I want to know if I'm exposed. If I take my PPE and I do things according to guidelines and I think I'm safe, but I want to know if I'm exposed or not. And then you do biological monitoring. And nowadays there are tests, urine tests available that you collect the urine of the workers and you analyze for the drugs, if you can detect the drugs in the urine. And it's actually the same way as it is done in the sports events, like if you have cycling or sports, that you check sports on drugs and things like that, if they are exposed or not. So this is actually the same way you can do it. So it's a little bit of difference between environmental testing or biological testing. This is what we do as a company. It's just for your information. We have 2 types of testing available on the market: monitoring surface contamination by wipe sampling. I will show that later on. And monitoring exposure of workers by urine analysis. So actually we can analyze urine of workers on 3 drugs: cyclophosphamide, ifosfamide, and a 5-FU metabolite, and by workers, which we do for some hospitals, and we also do for pharmaceutical industry. And then if you find positive urine samples, then you know that people are exposed or not. Then you know that people are exposed. The wipe testing, wipe sampling, we can do it on many drugs nowadays, but just for your information, that it's possible to do. How is monitoring with wipe sampling done? Well, here you see an example. It's on the left, on the top, you see a table where a preparation is done for cytodrugs. And what we did there, we took a wipe sample from this surface. I will show you here. This is the surface here. So they take it there. And then you see that it's transported by boxes. You have here a box where prepared bags are put in, which are sent to the wards. And then here on the left, you see here where we dripped the liquids on the surface. And here we did the wipe testing. I can show you later on how it's exactly working. So actually what you do, you define surfaces where you think they are contaminated. Then you drip the liquid on it. You take a tissue, you wipe it, you clean it actually, and then you collect it in a bottle. And then finally, with all the information, it's sent to the lab and you do the analysis. And then people get a report back. So it's very simple, straightforward. It's actually a way of cleaning how you do wipe tests. This is options for wipe sampling in the preparation area. So when you think about the pharmacy, then you have here, of course, the packaging. You have the vials here. You have the area where the pharmacist is sitting and doing some writing things, the desk. Here you have the cabinets where they do the preparation. And here you see also a little bit of these waste bins, which they flip, so that work that way. But here you have another one, which is a closed one. This is much better. And here is more or less a dirty area where a lot of drugs end up in the waste bin, the waste area. And here is a transport system. It's shipped, transport by, how do you call it, a system from the pharmacy. It's in the air, whatever, and it's coming up to the wards. And this packaging can be also contaminated inside. There's just an option about potential wipe surfing surfaces. When it comes to administration, then you have a little bit other options. This is, for instance, a table near a bed of a patient where they get their food, their dining things. Here you have some remote controls, which is the patient using. And when the patient is sweating, then of course he has sweat on his hands with the drugs, and it will be on the remote controls. Here the pumps are really one of the pieces which are really, most of the cases, contaminated. You will find always some contamination on the displays here and the touching parts of the pumps. Here you have a pole with the floor where you have the infusion. Often here is contamination, but even on the pole. So if you take a pole, have gloves on. The toilet, the most interesting place. I'll come later on it, but I can tell you already, never go to a patient toilet. Here we have some safety precautions, closed handling for waste. Here, another one too. So it's a sealing system that you put your waste in, and then it's sealed, So you don't be afraid to be exposed by inhalation of vapors, whatever. And here we have the laundry area where all the clothing is going and the lining of the beds and things like that. So just a few options where and around you can take some of the wipe testing. But there are many, many more options. As I said before, files are really risky if they are contaminated. And of course you don't know it and you don't see it. But in general, files can be contaminated, so be aware of it. And this is something that happened a few years ago, that they discovered that you can sleeve vials with a plastic film, which is seen here. And you see here the bottom of two ones. This one is not sleeved, but this one is sleeved. So actually there is a plastic film around the vial. This is very nice because it gives very good protection. So if the vial is contaminated in the production, and finally in the production they clean it, they wash it, but then you get the final step that in the end all the drips come on the bottom side because it's sprayed from the top and the water and the diluent is going down, but it ends on the bottom, and then it's a rowing system, so it can probably stay there on this side. But then when you take a film in the end, you cover the potential contamination on that vial, which can give up to 90-95% reduction in contamination. So if you can have these sleeved vials and you can buy it for your drugs, it can give a good improvement on safety. Here are two other examples. Oh, this is a study, we did on these vials. It's already a long time ago, 2005. But anyhow, it showed that when you have a standard decontamination of 72 vials, you find this contamination. And forget nanograms or whatever, just showing that there is contamination on these vials. And it wasn't, we checked for platin, so it's carboplatin vials. And then we did an improved cleaning on these vials, 75 vials, and you see it's already going down. So contamination on the vials went down after an improved cleaning. And then finally, when you put the sleeves on and we check 75 vials again, then you can see it's even going down more. So if you have vials with a sleeve protection, then you get a really good reduction of contamination. Here are two other options, or three actually. These are some vials with carboplatin, cisplatin, methotrexate. They are covered with the film, you can see here. And these have an extra protection, so if they are falling on the floor, whatever, you don't need to be afraid that they will break. There's an extra protection. But this extra sleeve protection is very good for reducing contamination. But anyhow, even these vials, touch it with your hands, not with your hands, but with gloves. We go back in the past a little bit, and talking about handling techniques for drug preparation. It's probably not really your business as nurses, but in the pharmacy it's the standard way how drugs are prepared. When we go back in the past, we had only the syringes, the needles, the bottles and the bags. And then I'm talking about probably 20, 25 years ago, maybe 30 years ago. And nurses who are close to their pension will probably recognize this, that this was the way they did it that way and at that time. And when you talk to them, they said, well, I did it this way, why do we need all these devices and things like that, I'm fine and so on. But anyhow, okay, there are nurses at that age now, they said, well, actually we did very bad in that time, it's better nowadays, so we improved a lot. But then, of course, industry is not really relaxing and things. They have always ideas how to improve safety and of course, sell products. And then all these type of medical devices popped up, like safety pins and devices to put on vials to protect yourself. So that was a really improvement already when you compare it to the needle syringe technique. And later on, the Lewis slip, which is a very soft connection, was replaced. I think it's now replaced everywhere, at least I hope so, by Lewis lock. A Lewis lock is a screw-ring, so you cannot really open it very easily. And then we got the protected vials. This was a new development about 15, 20 years ago. And the last 15 years ago, we have the closed systems coming on the market, so that's already a new step forward. And more recently, actually, you see a lot of robots doing the work of the pharmacy technicians. And Well, one of the problems is with pharmacy technicians who do preparations that they have a lot of problems with the functioning of the pulse and all these bones here. And if a robot can do that work, and that's of course an improvement for them, and they just control the robotic system by working on the computer and put the things in, take it out. So it's for them more relaxing when it comes to physical job. So a lot of things happening actually the last 5 to 10 years. And well, we will see what the future will bring. I see not really new things, but you will never know what will happen. Now we go back to wipe testing surface contamination. And this is just an overview. As I said, wipe sampling is a very simple process. You can take a wipe test from every surface when it's rather flat. So you just spray a liquid on it, you take with a tissue, you clean it, you wipe it, you collect it, you send it to the lab, and they will analyze it. And this is an overview about the studies we did in the 90s. So it's one of these is done by Tom Connor and myself in the US and Canada. It's already a while ago, 99. So it's really one of the beginning studies. And actually what we discovered here, when you go to the pharmacy and you check the area on preparation area with cyclophosphamide, one of the drugs, but you can do it with many others. And then when you go in this area and you check the preparation surfaces, the floor before under the cabinet, preparation room, the floor somewhere else in the room, tabletop and things, but also the corridor outside of the preparation area. And then what you see is there is contamination everywhere. Just forget the figures, I will come on that later, but what we see is contamination. Wherever you go, if you go to Canada, US, Europe, Netherlands, Japan, you will find it everywhere, and I'm sure you will find it here too. That was a long time ago. We do still these studies. I come later on that on very recent results from the Netherlands, about 1,000 samples we analyzed there. I give you some recent information on it in the Netherlands today. Here is a study overview 2007, 2012, and it's in administration area, so where the drugs are administered. This is Dutch hospitals, just to give you an impression. So when you go to the floor near the bed of the patient, the armrest where the infusion is given, the floor in the area of the cyto waste bin, the floor where the infusion standard is standing, the toilet of the patient, cleaning area, a table where cyto is used, but also office areas and relaxed room of nurses, so where they have the coffee and their breakfast and the meals and things like that. Anyhow, you see cyclophosphamide was detected everywhere there. 5-FU, not that frequent, but anyhow it was there, and platinum drugs were also detected. So actually also when you go to these areas where the administration where drugs are given, you see a lot of the positions that appear to be positive. And this is a 5-year study in quite a lot of hospitals in the Netherlands. So also nursing, there is contamination. I will come back later on this. When we did the first wipe testing, and we developed these tests, and the first one were done in around 1990, so it's about 25 years ago now, so it's quite a long time ago. 1990, some of the first tests were done, and we had not that sensitive equipment we have nowadays. At that time, we were very straightforward with our techniques. We had HPLC, GC. I will not talk in detail about this, but actually at that time, we detected quite high levels. As you can see here, it's micrograms on a square centimeter. That was the level we could detect in that time with these techniques. But then new techniques came up. The labs were getting more sophisticated, better equipment, higher sensitivity. So 10 years later, we were able to detect the nanograms, so 1,000 times lower detection than we had in the 90s. And in 2010, let's see this period where we are living now, we are on the picogram scale. So we can detect 1 million times lower than in the 90s. And, well, the future, we don't know. Maybe we can detect 1 molecule on a square centimeter. I don't know, but you never know. The point here is if you are able to detect very, very low amounts, then the number of samples, positive samples, will increase, because the lower you can measure, the more positive samples you will get. So when you have a result of a study showing that 100% of the samples is positive with this detection limit, then people say, well, everywhere, but what does it mean when I have picograms? What is picograms? You cannot see it. It's one drop in a basin or whatever. So what does it mean? And said, well, and you find it everywhere. And yeah, but what is the relevance of this? What does it mean in terms of risk or exposure or whatever? So we had a big debate starting in Holland many years ago, and said, well, especially the pharmacists and the nurses said, well, we want to work safe, but we want to have data that really prove that we are safe. So if there is total contamination on picogram, but there is no contamination on nanogram, well, makes it sense. What does it mean? So we had a big debate. And we had a debate of when we do these tests, which were at that time already obliged in Dutch hospitals to do once a year. And said, well, if we find always contamination on a picogram, we want to know what does it mean. Is there really a risk? And then we had a big discussion with pharmacists, nurses, hygienists, and toxicologists about the impact. What does it mean in terms of risk? And we said, well, what does surface contamination mean in terms of risk, and what level is acceptable? And actually we decided we accept contamination. As soon as the longer the detection limit is going down, we can measure smaller and smaller amounts. So there will always be contamination. And we accept it, but the question at that time was, what do we accept? What is the level which we think is acceptable in terms of risk, and which is the level which is not acceptable? And then we had a very pragmatic approach on this, and we said, well, how do we do this? And we do it in a very simple way. Let's go back. Yeah, this one. And it's quite very simple. We said, well, we have to deal with 50 drugs, probably. We cannot test all the drugs. It's too complicated. We have no way to do it. There are no tests for all these drugs. So we take the most worst drug. And that was cyclophosphamide, as I said before. Cyclophosphamide is very quick penetrating your skin. So if you have it on your body, you can be exposed very quickly. It's very persistent drug, so you can only remove cyclophosphamide by cleaning. So when it's in the environment, it will evaporate, it will stay there. So you can only remove it by cleaning. And we have good tests to measure this drug. And then we collected all the data from all these wipe tests, and we discovered actually that 90% of the wipe samples showed this level, 0.1 or lower. And 99% was below the 10. And the point was that we also included urine testing. So when we did this wipe testing, we also checked the urine of the workers who are working in this area. And we discovered that no positive urine samples were found if the contamination was below this level. So nobody was exposed when the contamination was at that level or lower. And then we said, well, this is a very good start. That we said, well, this is an acceptable level because it doesn't turn out to be that there is a risk, because we cannot measure the drug anymore in the urine, so there is no relation to any risk anymore. And that means we consider 0.1 nanogram on a square centimeter as a safe level. And of course, you can debate a lot on it, but the point here is too, if you take, for instance, other drugs who are not so quickly passing the skin, then of course this can be a little bit higher probably. So this is really a worst case scenario. So we said, well, this will function for cyclophosphamide, then it will also function for all the other ones because they are less toxic. They're of course toxic, but less toxic. And you are not so potently exposed to this drug. So that's the reason why we selected this 0.1 as the reference. So all contamination tests have to be below this level. And then we said 10, which means 2 magnitudes higher. is unacceptable. That's really too high. When you have all these high figures, then a lot of positive samples appear, so really people really get exposed. And we said 10 is really not done. And in the end, it turned out to be in this model. And we call it a traffic light model, and it's very simple actually. Traffic light said green is green and go on, nothing, no problem at all. And red is stop. And that means if you have contamination below 0.1, then you're on the safe side. Oh, you have always to take care, but you're on the safe side. But when it's 10, then it's stop. It's really too much, too high. We know that people are really getting exposed by this level of contamination. But in between, it's a little bit, what shall you decide? So we split it up in two parts, yellow and orange. And yellow is actually indicating, well, okay, there's something going on, take care. But orange is indicating, well, you are close to red, so really take care now. So be sure that it's not increasing, so really, do something. And actually the actions we put on these colors are very simple here. Green is that you check once a while, which is more or less once a year. And here in between we said, well, you have to make an estimate. If you have yellow results for a few samples, then it's probably not that problematic. But if you have a lot of orange results, then I think you should do something by making an estimate, try to improve situations, look where are the problems, where the potential problems are, and try to take some measures, but follow up with another test to check after a while, 3-6 months, if it's the same, increase or reduce. And of course, the measures you take should result in a reduction of the level. So actually the aim is from here to go down to green. Red is totally not done. Red is really too big, too much, and that means really stop and clean, very, very good cleaning, and then check again and go back, and also check how you handle, how you do the preparations, and how do you the administration, because this is not really acceptable. There's really something going wrong there. This model is published already in 2011, but it's updated last year in November, and you can read it in www.doctorwho.nl. It's in Dutch, but I think you will manage. I think you will manage. You will manage. But this is the standard we use nowadays in Holland. So the standard is that testing in Holland, in hospitals, wards, pharmacy departments is done every year in all hospitals, and the aim is that the level should be in the green, on the green side. And it's an obligation in that way that, people agreed on doing it this way, and the Inspectorate of Labor is not checking the results of the testing, but they only check if the tests are done. And then it's up to the hospital to take the decisions on the results. So if the Inspectorate of Labor is coming in a pharmacy or nursing department in a hospital, whatever, and they check and there are no testing results available over the last year, then you get the option to do the testing. If you don't do it, then you have a problem. But they don't look in the results. If the results are very bad, they do nothing. That's the situation. But I think having the monitoring every year as a check, I think that works fine. Okay, I have shown you the results of the studies before, in a few slides before, on the preparation in these countries, Canada, US, Europe, Netherlands, Japan. But now I put the colors in, so you get a better picture of what it means. And then you see where the problems really are. So if you go to the Cyto, the tabletop of the Cyto preparation in the cabinet, where they do the preparation, actually where the most risky things are done, then you see you have the red and orange colors there. So in the preparation area, and of course you have to consider this is a while ago, so probably when you update it a little bit, it can be a little bit different. But at that time, there was a lot of contamination, and that means actually that contamination is moving from the preparation on the bags and finally ending up in the ward. Here you see when you are a little bit more away from the from the preparation, so the floor inside the room, then it's reducing a little bit, but anyhow you can have a lot of contamination there too. But even outside of the corridor, so where everybody is walking, you find some contamination there. But it's lower, quite lower compared to the other places. This is the other one, the Dutch hospital, 2007-2012. I presented a few slides before, but now also with the colors in. And here you see the positions close to the patient. So that's really orange. So there's really something going on there. But here the toilet of the patient. As I said before, never go to a patient's toilet, but this is really showing never go there. So there is really a lot of contamination. And of course, the patient who has the drug in his body is going to the toilet, is being there, sitting there, and there can be splashes and whatever anywhere. So it's reasonable that it's there, but it's not good for normal people versus going to a toilet where patients have been. So split it up in separate toilets. But here in the coffee area, the relaxed room, you find drugs. So be aware of it, that it can be spread everywhere. So contamination can be, and these were 3 drugs we tested, so it can be actually everywhere. And this is quite recent. As I talked about frequent monitoring, this is a hospital in the United States. It's just giving an example. There are many examples to present. But this hospital, they do every half-year monitoring on 12 positions, the same positions every half year. And you see here it's oncology department, pharmacy, nursing, and another pharmacy department. And when you do this and you put these colors in, you get a very nice overview where you have the main problems. And the main problems are here, the preparation area, so the pharmacy here. It's a little bit going down here, but anyhow, sometimes you have some positive spots. And here, the floor by the hood appeared also to be a little bit, contaminated, more or less every time. And sometimes it depends a little bit higher here, a little bit lower there, but anyhow. But this monitoring, when you do it on a frequent base every half year, it keeps your awareness still there. And when the testing is done independently, unannounced, just people go in, do the testing, and then you can have this as an overview. And I think this will work well, because it will very quickly appear where the problems are. And then you take your measures, repeat the testing again, and then hopefully it ends up well. So this is how monitoring can be done. And this is only the white testing. So I did another study the last 5 years, actually a quite big study on surface contamination in the Netherlands, 2015. It took about, let's see, close to 2,000 wipe samples. This is quite a lot. 16 hospitals were involved, about 700 in the pharmacies, preparation, a little bit more than 1,000 in the wards, clinics. And this is what we have discovered. 1,280 samples on cyclophosphamide, platinum drugs, methotrexate, and just a few were for ifosfamide and 5-FU, but most were analyzed on these compounds, the platinum, methotrexate, and cyclophosphamide. So, what we discovered from the study was that the positive samples was about 30% of the cyclophosphamide ones were positive, 25% were for platinum drugs, 17% for methotrexate, and none for 5-FU and ifosfamide. The very high ones above the 10, so that's the red color, that was 6 for cyclophosphamide, 5 for methotrexate, but it appeared in the end that most of the contamination was caused by production of capsules. And sometimes for pediatrics, for kids, doses have to be adapted, and you cannot use the standard tablets or pills or whatever. So they have to produce by powder, by their own. And that's mainly for methotrexate and for cyclophosphamide. So it really appeared that there was a lot of powder appearing and leakaging during the preparation. But this is the area we are interested in. So that's more the yellow and orange area. So then it appeared that there are quite a lot of positive samples appeared. But now we go more in detail. This is the overview. Most of the samples were below the 0.1, are not detectable, and just a few in this area. And I said here are the two high ones, the methotrexate, the cyclophosphamide. So most of the samples were below the 0.1. Now we go in detail, and this is interesting because here are the positions that were measured in most of the hospitals. So they're all identical indicated. So if you go to the cabinet, the biological safety cabinet, the walls, so inside, the surface, underneath the grill, so it's all inside the cabinet, but you have to share the floor, objects outside the cabinet, the waste bin area, transport equipment, objects and positions, non-sterile. And then you see really where the problems are. The problems are here in the cabinet. The cabinet is showing the highest percentage of positive samples. And the positions non-sterile, when I specify this, these are mostly vials, so outside of vials that are contaminated. So before they go into the sterile room, they are checked, and we check them, and quite a lot of these samples. You can see more or less 50% of the vials were contaminated on the outside with the drug. But indicating here, again, proven after many years that inside the cabinet, here are the problems. So really the preparation itself causes spillage and leakage, and that will always end up in the final product. So here the overview. 65% positive, 41 here. This was the vials, as I said before. 33%, 26. So actually a lot of positions in the pharmacy close to the active handling were contaminated, and the vials. So actually really a lot of vials were contaminated. Now we go to administration, so nursing area, and outpatient area. And here we have to deal with infusion, patient room, bed and chair, floor, toilet of the patient, knobs for door light, dirty room, toilet of the staff, area of nursing staff, and medication room. And here again it appeared the toilet is very predominant here, floors and bed close to the patient, the shares. So actually where the active administration is done here. So really close to the patient area, the administration, there is the contamination. But anyhow, here quite a lot of samples, so over 50% show to be positive in that yellow-orange area. Well, here the overview of the study. 25% was above, was positive, and here 59% patients area, the floors, the toilets here, bed, chairs, 52%, and then the other objects, waste, don't forget the waste here, and the toilet of the staff. So even the toilet of the staff was contaminated with the drugs. I think the last has to do with bed cleaning. What you often see is that cleaning is outsourced, done by people who clean as they clean a probably normal house or whatever. So they have no clue when they have to deal with cytodrugs how to manage this. And what you often see is they start cleaning the toilet, and then they use the mop and the water for cleaning the rest of the rooms, and they don't clean, they just spread it. So you have really to start at the areas which are not highly contaminated. So start in patient rooms and in outpatient areas, and end up in the most dirty places like the waste area and the toilets before, or change the water more frequently or things like that. So often it's just logic thinking, but you can cause a lot of contamination if you do it in the wrong way. So most of the positive positions are located close to the patient. Contamination on toilets of staff used by patients. Number of positive samples in administration areas are higher than in the pharmacy. And this has all to do with the fact that you have to deal with the patient, not with a simple vial. That is wipe testing. So how we do a wipe testing, what you can do with the results of wipe testing. So they can be very illustrative when you show people with the testing. You don't see the drugs, but they are there, and that you can discover where the problems are. But then we go a little bit more to the work itself, because when you have this result, a lot of people say, oh my God, it's everywhere. Can I work in a good way here, and so on. And of course you just go on with your work, but realize things that something can go wrong in this process or in this process. So it's a little bit awareness, and always realize when you work in a team, and I've seen it many times, there can be 9 people in a team who do it perfect, and you have one doesn't care, and then it's a problem for everybody. So you have to teach and educate each other, so that's really relevant. Okay, that was wipe testing, urine testing, testing exposure, testing contamination. Now we go to question 5. And I will focus on closed system drug transfer devices. And the reason is, as I said before, this is the highest option in your strategy you can follow. The best option actually to stay away from the drugs and to be exposed. Okay, closed system drug transfer devices in general, there are many today, but are devices to prevent chemical contamination, drugs to go out, particulate contamination to get in, microbial contamination to go out, all of them or none of them. Okay. Okay, let's see. Oh, interesting. Okay, chemical contamination to go out is 24%, and 74% said all of them. Interesting. But actually, its answer is number 4. A closed system means nothing going in and nothing going out. So no drugs going out, no particulates going in, and no microbiological things going in. So it's totally closed from everything. So the first answer, chemical contamination to go out, that's of course okay. But it's also that nothing can get in. Good. This is the definition of a closed system transfer device, a definition set by the National Institute of Occupational Health, NIOSH, in the United States. I took this definition. There are some others, but I took this one as an example, which I think is very commonly used. But as you read the description, then it's a lot of wordings to give that definition. And it's in fact that a closed system transfer device is a drug transfer device that mechanically prohibits the transfer of environmental contaminants into the system and escape drugs, as I said, getting out. So that means actually a system that is airtight and leak-proof. That's roughly a definition of it. Okay, now we get another question on closed system transfer devices. CSTD, as protector of the product, the drug, regarding microbial contamination, enables you, 1, no need to use a biological safety cabinet or an isolator, no need to use personal protective equipment, no need to work in a controlled environment clean room, all of them or none of them. Okay. Wow. Oh. Yeah, that's a really good answer. You always need the extra things when you use a closed system, because there can always be a mishaps, an accident, or whatever. And for that, you really need to have the other things in place too. So it's the other things you need also too. So really, you need them all, 1, 2, and 3. Good. Yeah, surprised that you had that good. Okay. Okay, another question about closed system transfer devices. CSTD as protector of the product, drug, regarding microbial contamination enables you, 1, no conservation of the drug, 6-hour conservation of the drug, 1 day conservation of the drug, 7 days conservation of the drugs, or no limits on conservation of the drugs. So if you use a closed system, can you keep it on a microbiological way for a longer period than 1 day, 1 hour? Or is it not possible at all? Okay. Interesting. Well, there are two opposites. One saying no conservation on the drugs. And there is a group who's saying, well, no limits on conservation of the drugs. So that's really the opposite of each other. Well, to my opinion, the answer is in between. No, it's a debatable question. Therefore, I put it in. But there are studies showing that you can use a closed system drug transfer device for 7 days, saving the drug on a microbiological way. So it's possible. But there are some restrictions on that too. So, for instance, if you use vials for preparation today and you put a protective system on it from a closed system on it, you can use the drug the next day or the day after for other preparations if you want. But that's only when you have a CSTD on place. So it's possible to save it. And there are studies showing that it can be up to 7 days now. So you can do it. The question is it allowed or not? That's another issue. There are probably rules here in the UK, I don't know, saying you can never use it. You have to throw it away after 1 day or whatever. But it's, from a microbiological point of view, it's possible to save the drugs for a longer period if you link it to a closed system transfer device. Of course, there is another thing, that's the chemical stability. That's another point. Of course, it can be that the chemical stability is saying of a drug, you cannot use it for the next day or 2 days because it's very unstable. That's another point. But when it's coming to microbiological things, then there are things, studies showing that you can save it up to a longer period. But it's nice to see 55, 25, so it's really good. I have another one, also on a CSTD. And that has to link with that you can keep the drugs for a longer period. So if you can keep a drug for a longer period on a microbiological view, then you can use it the next day or the day after and so on. So therefore this question, using a closed system transfer device for multi-dose vials, so that means that 1 vial can be used for 2 patients, saves money including cost of CSTD. How much as percentage of the budget, taking in total the budget for drugs preparation, oncology preparation, how much is that? Is it 0%, so there is no savings? Is it 10%, there is a little savings, 20%, more than 30%, or no savings at all? So there's extra budget needed because CSTDs cost money. Up to you. This is also a little bit a tricky one. Oh, interesting. So there is a group of people saying, well, very small ones said 0, so there's no saving on the budget, whatever. So whatever you do, you gain no money on it. And 15%, there are no savings at all. You need extra budget, so it costs money, actually, totally. Well, the answer is in between. And the question is a little bit misleading because I didn't mention what drug vials. But that's the debate, of course. If you just focus on cytotoxic drugs, then you can probably save 10% on your budget, including the closed system transfer device. If you use a vial, the remaining of 1 vial for the next patient. So actually, that means that there is no drug. You don't waste drugs. You use all the drugs if you can. So no waste of drugs. You can save 10% on the most cheap ones, let's say 5-FU, cyclophosphamide, these things like that. But if you also apply them on the monoclonals or the more expensive ones, then you can save certainly 20%, but probably also 30%. So you can save quite a lot of money if you apply the closed system on especially also the monoclonals. So if you take the total package of the hazardous drugs which are prepared in a pharmacy and administered in nursing, then there is a lot of money you can save. So the debate about that closed systems cost money is, I think, is not an issue, because you can save money when you use the drugs for more patients, and especially the expensive ones. So it's, I would say, a lie saying that closed systems are expensive. And in this way, you save drugs, you can treat more patients, you protect yourself better, and you save money. So that's all in the issue. Okay, then we come to another one. Question 9. Also about closed system transfer device. Effectiveness of a closed system transfer device should be checked by. So there is a lot of debate, what is a closed system transfer device or what not. But you have to do some things to really show that a system is a closed system transfer device. But the question is, what should you show? Is it 1, a leak test and an air tightness test? So there is no leakage and it's a tight system. A microbial ingress test. 3, measurement of chemical contamination in the environment and workers' exposure. 4, 1 and 2. Or 5, 1 and 2 and 3. Okay. Well, the majority is convinced that you should do 1, 2, and 3. And the smaller group is saying number 4, 1 and 2. Well, to check a CSTD, it's of course not needed to do chemical contamination and exposure tests. It's nice to do, but it's not needed to prove that the system is closed or not. It's nice, but not necessary. But anyhow, you should do a microbial testing and a tightness and leak test. So the correct answer here is answer 4. I like that you answered number 5, but it's 4. Okay, I will show a little bit about closed systems. We have done a study a while ago in the United States. Closed systems on the market at that time, Facile was a very predominant closed system on the market. It's now produced nowadays by Becton and Dickinson. And we did a study in the 22 hospitals on 4 surfaces, 4 standard surfaces. And it's indicating here the cabinet, biological safety cabinet surface, the airfoil, the floor in front, and the counter where all the prepared products are put on. And what we discovered here is that when you take cyclophosphamide as a drug to test, and we do the wipe testing on these surfaces, then you see here, with the standard technique at that time, which was just a very simple syringe and needle technique, you have quite a lot of contamination here. You see it here, especially on the airfoil, so the area where the real preparation is done. And when you replace it by Facile and retest after a while, you see a total reduction. So very, very good reduction over time in these 22 hospitals in that period. And then we repeated this one again. 30 hospitals, 5 years later, this period, same surfaces, same positions. And again here with the standard technique, needle syringe, which was still used in the United States. You see here the results again, and with Facile you get a significant reduction. So actually it's showing when you use a closed system, you can reduce contamination very quick, if you're going down. And when you compare these two, and you just look at the colors, then you see that it was really not so really much difference. And it was strange because in between that period, when we did the second study, the United States developed or actually updated a new guideline on how to handle cytotoxic drugs. And that guideline was implemented, a new guideline, in these hospitals. But then it appeared no difference between the two studies. So actually, new guidelines with a lot of paperwork doesn't mean that you improve your situation. You have always to validate it afterwards. And this test is actually showing that all these new guidelines didn't make any sense at all. Nothing has changed. This is just an example of closed system transfer device. Later on, I will present another one, but there are a few on the market nowadays. I come to conclusions and recommendations when it comes to the antibiotics. We don't know that much about antibiotics, but in potential, they are very potent drugs. So you have to take care of them too. And the amounts, the number are much higher than for the cytodrugs. We know that there are probably some effects, but we don't see them really so much as reported in studies. But as the number of workers is quite high and effects may be delayed, there can be a potential risk here. Skin contact is very relevant here. Inhalation is an issue when it comes in areas where there are no ventilations or bad ventilation and no cabinets or isolators. Gloves have to be worn. Vials, the same issues with the cytodrugs. And what we have seen in many places now, that the protection of the workers to antibiotics is very severe when you compare it to the cytodrugs. And so potentially the exposure can be substantially higher. No, but we cannot measure in the drugs the antibiotics nowadays, but probably in the future. So we have to focus on the wipe sampling now. The Hungarian study has been very, I would say very good in that way, that it's really showing where the problems are and that people can be exposed and that you have environmental contamination. But you can do a reduction substantially with a closed system transfer device. Here again, open connections don't do that. Closed systems reduce it. No patients' toilets here either. Surface cleaning is very simple for antibiotics compared to cytodrugs. You can just clean with water. You remove antibiotics, no problem at all. And I think we have to consider that more monitoring is recommended because we have no idea about the real exposure to these drugs for the many people who are involved, which appear to be about 11 million nurses in Europe. Take-home messages. Just a few. Cytotoxic drugs and antibiotics may cause adverse health effects. Put that in your head. That's the basic of all. Protect yourself, but also your colleagues, according to the EU strategy. That means first, close handling, second, ventilation, third, personal protective equipment. So if you're now on 3 or 2, consider really to go to level 1, that level of closed handling. Third, use gloves whenever there is a risk to get in contact with the drugs. It's really very important. Vials, including the packaging, prepare drugs, waste, don't forget the waste, bed linen, clothing and the urine and all the things from the patient. Never use a patient toilet. Keep that here too. And the last one is more a recommendation, but I said, well, ask your employer to keep you updated on this issue. So be informed, be trained. Request for continuous training, monitoring, wipe sample, and or urine testing, because then you know really what's going on. That's my last one. And that's me in PPE, not in a pharmacy, but in pharmacy production where the drugs are produced. So that's the precaution you take there. So thank you. There are probably some questions. Are there some questions?
When you said about protecting from exposure to patient fluids and things, is there any kind of idea of time? How long we should be informing patients that they consider to be giving out contaminated fluids?
Yeah. In general, there are some drugs who are excreted very quick, so let's say within 1-2 days. But for instance, if you take, I think, cisplatin or carboplatin, that can take up to 6 days before it's totally leaving the body of the patient. So, if you want to do it in the same way, I would say 6 days, but that's probably too long. So maybe you can decide for cisplatin that, but for the others maybe 2-3 days, roughly. Yeah.
In our potential brave new post-Brexit world, do you envisage that the legislation in this country is going to be adversely affected? Because I worked in the NHS pre-COSH directives, and most of the legislation we live in now is due to EU legislation.
So actually, when I understand you right, you ask me what do you think about the legislation in the UK now?
Do you think it's going to be affected by us leaving? Do you think our safety is going to be the safety of?
I think it could change. My impression is that the UK is not really in front of labor issues when it comes to protection of workers, whatever. That has ever been within Europe too. I'm not sure what will happen if you leave. I think it can probably be even worse. My impression is that the UK is not really in front of this. But the initiative by the EU member of Parliament, which is a British man, he is really effective when it comes to labour, things like that. So he's really in charge of it. But yeah, over 2 years we don't know. So yeah, but you can do it yourself. So you have to form a collective and work together to press things going. Helen.
Hi, Paul. Quick question for you on the antibiotic study. Did you consider the innate stability of the drugs that you were testing? Referring to the antibiotics, some of them are very unstable, and the meropenem, which wasn't detected, that's a very unstable antibiotic. Do you think that might have contributed to you not detecting that?
We evaluated all the wipe tests on the antibiotics in our lab on storage, temperatures and things like that. Our impression is that the most of the drugs are rather stable for a long period, except I think it was piperacillin, which can be a little bit of a problem. But all the others appear to be rather stable for a long time, as we tested in our lab. So that's my impression. But anyhow, if it's less stable, that means that whatever you detect is always an underestimate of the real contamination. So that means that the figures will be anyhow higher than presented here. So that makes the situation even more worse.
Thank you.
You very briefly touched on monoclonal antibiotics. Now that they're given subcutaneously, they're prepared more in the nursing quarters or nursing environment. Is there any measure on what vulnerability nurses are exposed to with monoclonals?
No. The point is that the analytical things to measure any contamination, exposure, whatever, is very problematic when it comes to monoclonals because they are very complicated molecules. So that's not so simple to do. So as far as I know, there is nothing published on that. The only thing what I know that there is a debate going on what type of precautions you should take for when you are handling monoclonals. And some people say, well, because these molecules are very big, the chance to be exposed is very small because they cannot enter your body by inhalation or by skin contact. But there are some drugs appearing already, some publications showing that some of these are really showing carcinogenic effects, and effects on reproduction. So I think we should apply the same strategy on protection for monoclonals than for the cytodrugs we have nowadays. But there are no testing at this moment.
Thank you.
More questions? Then we go to the coffee break. Thank you.